Thanks for the invitation. Um, yeah, and I haven't been able to attend these, unfortunately, because last semester and this semester, they occur at the same time that I teach. And I was able to do today because this is my fall break. So, oh wait, what's happening here? It's like going through my slides somehow. Okay, now why is it automatically doing that? Okay. All right. Okay, so I'll be talking about model selection for trees versus networks using a form of cross-validation. And this is work with a PhD student of mine, Rashid Al-Hassan, at the University of New Mexico. Okay, well, so I'll briefly talk about phylogenetic networks. Not that this group needs much introduction on that. And inferring networks is rather difficult. So a sort of easier problem to work on is, given a small number of candidate networks, here I'm just going to use two, can we distinguish between them? And sometimes we might want to distinguish between a network versus a tree. Okay, and there are different approaches to that. So I'm going to be looking at cross-validation as an alternative to other things, such as using AIC or hypothesis testing, for example. Okay. All right, so here's an example of a network and a gene tree evolving within the network. So, so the usual assumption here is that the network features are at the species level and gene trees are trees within the network. So the gene trees are completely tree-like. And we have multiple loci, so each locus can be a different tree within the network. And one thing to consider is we allow gene trees to not necessarily be displayed by the network. So this is an example of that, where the B and E lineages in the gene tree go separate ways at this hybridization node. And so you end up with a gene tree that's not displayed by the network. Okay. Okay, so so species networks can predict topologies for gene trees that they lead to probabilities that are things that you might not be able to get under a tree. And since they predict different probability models, we would hope that we could distinguish between tree-like networks versus non-tree-like networks. So this is just an example of a couple of probability calculations for for gene trees given these two networks. Yeah, in this case, here are two gene trees that have identical probabilities under this gene tree, but they don't have identical probabilities under this network. Okay. Okay, so given a collection of gene trees, we'd like to infer the correct network, which might be a tree. Okay, so a number of challenges. The space of possible networks is much larger than the number of tree topologies, so that makes searching over the space quite slow and difficult. The space is not clearly defined, so some algorithms or inference software restrict the problem to level 1 networks, which don't have overlapping cycles, for example. But I think everyone would like to work in a more general setting than that, but it's but that makes things more tractable. Likelihood calculations are much slower for networks than for trees, partly because there are more parameters. And the parameter space is kind of strange. So different networks have different numbers of parameters, which from a statistical point of view, it makes the problem seem a little more like model selection than estimation. Although, I remember seeing papers in the 90s that argued that just regular phylogenetic inference should be thought of as model selection as opposed to an estimation problem. But that seems even to me that seems even more natural to think about it that way for networks as opposed to trees. Okay, so less ambitious than trying to infer a tree from the space, or infer a network from the space of all possible networks, is to try to decide between a small number of candidate networks, or even two of them. And I'm going to use an example of using cross-validation to decide between candidate networks. So cross-validation was proposed in this paper by Knockley in 2014. So this is where they introduced using branch lengths in the gene trees, which I'm not going to do here. So I'm going to use just gene tree topologies. I would like to extend this to trying this with branch lengths as well. So my experience with using branch lengths in the gene trees is that some methods, especially the type of likelihood method that they're using here, will be extremely sensitive to errors in the estimated branch lengths in the gene trees. Okay, so in their article, they use cross-validation by predicting gene tree topologies for different fitted models and finding which network does the best job of predicting gene tree topologies, gene tree topology frequencies. So they're using branch lengths in the gene trees to infer the parameters in the network, but then in order to do the cross-validation part, they're using the network to predict how often different gene tree topology frequencies would occur, and figuring out which network does the best job of predicting the gene tree topologies, not the branch lengths in the gene trees. At least that's my understanding of the paper. Okay, so a concern for this approach is that the number of possible topologies grows rapidly with the number of leaves. And as a result, for larger trees, all topologies might have very low expected frequencies. And in this case, it might make sense to summarize gene trees into larger categories. So that's kind of the motivation for this paper. I had another student who graduated earlier this year, and we looked at, given a sample of gene trees, how many distinct topologies occur, which is kind of a fun problem. And if you have a sample of 1,000 gene trees, is it possible that every gene tree has a unique topology, for example? And when does that occur? When is that sort of expected to occur? So it's possible that every tree topology has an expected frequency of less than 1, for example. Anyway, so an alternative for using cross-validation on predicted topology frequencies would be to use rooted triple frequencies. And the motivation for that is that there are many fewer rooted triples. So as a result, even if every gene tree topology is unique, the triples are not going to be unique. And so you'll have higher counts in the different categories. So for 8 leaves, for example, you have over 135,000 rooted trees, but only 168 possible triples. And of course, this could be done with quartets instead of triples, but for this project, we just stayed in the rooted setting. Okay. All right, so the network we used was this one. So it only has one hybridization event. And one thing I was kind of interested in is, can you improve inference by having lots of descendants of a hybrid? And so we took this as sort of the largest network. Oh, sorry, it says 2 hybridizations. It should be 1 hybridization. Okay, and we're just distinguishing whether this hybridization occurred, whether the network has this hybridization event, or whether the data come from a tree. And so we set our inheritance parameter for the hybrid node to be either 0, 0.1, 0.2, or 0.3. So if it's 0, then the data is tree-like, and these taxa B, C, E, F, and so on would cluster with A, more likely than with D. And as you increase gamma, there should be more signal for hybridization. Okay, and the numbers here represent branch lengths. And yeah, okay. And so we used different subsets of the number of descendants for the hybrid node H1. So for the smallest case, we had a network with 4 leaves. So that would be A, C, B, and D. So A, B, C, and D. And then we increased the number by 1 at a time. So A, B, C, D, E, A, B, C, D, E, F, and then all the way up to 9 leaves. So the number of descendants of the hybrid is between 2 and 7. Okay. All right. So for the simulation study, we used 1,000 gene trees. Well, for each setting, so 1,000 gene trees simulated from the network for different values of gamma, the inheritance parameter. The 1,000 gene trees are partitioned into 5 sets of 200. So this is to do 5-fold cross-validation. So each each fold is going to have 200 gene trees. So that's used to create a test set, and the remaining 800 loci are used for training sets. Okay, so for each training set, the network parameters are optimized. So this was done in PhyloNet. So the topology is fixed, and the network parameters, including branch lengths and inheritance probability, are optimized using likelihood of the gene tree topologies, and that's likelihood on the full network, which is rather slow when at least when you have 9 leaves. Okay, so then topology and rooted triple frequencies are predicted. They could be predicted theoretically, but we just did it using simulation. So simulating from the optimized network, and then compared to the frequencies in the test sets. Okay, so then you have 2 networks, N1, which is the true network, and a tree, which is basically the tree you'd get if the hybridization parameter was set to 0. So then we're comparing those, and we're producing a score for each network. So the score is the sum of squares. So we're summing over all the folds and summing over each gene tree. Well, actually, I is going to be the I is actually indexing the topologies that are observed. So okay, so O sub ki is the observed frequency of category I, so meaning topology I. Um, well, okay, so if we're using cross-validation using full trees, it's going to be a topology. If we're using triples, then I is going to represent one of the triples that's observed. Okay, and we look at the squared deviation from the expected frequency, where the expected frequency is estimated using simulated data. And this could have been done using theoretical probabilities. That would be possible too. But it's sort of in the interest of scalability, it's faster to simulate these things than to calculate their probabilities, especially when you have, especially if you have a nine leaf network. And ideally, we'd like an algorithm that will scale to larger cases. Okay. Let's see. Okay, so depending on the score, you choose which network. So whichever network gets the lower score, that indicates a better fit. And I guess I should indicate, other variations could be used. So if we divided by the expected value, we'd basically be doing a chi-square criterion here. So we'd be essentially doing some sort of goodness of fit with a chi-square. So instead, we're basically doing least squares instead of chi-square. So we haven't tried both of those. I wouldn't expect them to come to radically different conclusions, but that would be a variation to try. Okay. All right, and instead of computing expected frequencies for all possible gene trees, we only use those in the data, in the simulated data. So instead of summing over all possible topologies, we're just using the ones that we happen to find. Otherwise, the sum would just have tons of zeros in it, which would be okay too. Okay, and the expected values are estimated by simulating some number of gene trees. So here we used m equals 200, which is the same number as the test set. So the idea is you have observed data, and which is the test data, and then you have the expected frequencies, which are simulated from the inferred network. And you're trying to compare and see how similar the observed values are from data that's simulated from the network. Which is like a parametric bootstrap. Okay. Let's see. All right, so that's the approach, and then for this network, we had between 4 and 9 leaves total. So 4 leaves means 2 descendants from the hybrid, 9 leaves means 7 descendants from the hybrid. So in these plots, the open circles represent the proportion of correct decisions, classifications based on cross-validation for the triplet approach versus the full tree approach. So for 4, 5, and 6, we see very similar performance. And the advantage for the triplet approach is more that it's scalable, or better scalable than using the full trees. So even just the summarizing the data to figure out the observed frequencies is faster using the triplets than using the full trees. So they had similar performance there. And then for 7, 8, and 9 leaves, the triplet approach is starting to have better performance. I guess one thing I was kind of hoping to find would be higher accuracy with more descendants of the hybrid node. And we do see that with 9, but it's not a very obvious increase in performance with more descendants of the hybrid node. So each of these points is an average of 100 iterations in the simulation. And so we could maybe get a little more clarity by doing a larger number of iterations there. Anyway, so I'd see this as kind of a proof of concept that using cross-validation with the full trees just doesn't seem scalable to me, because it's very likely that you'll just have a bunch of unique trees, and counting how often each one occurs, you're just going to get zeros and ones for the most part. So to me, it seems like you should have some sort of summary of the data that isn't going to have that problem. All right, so some motivation for thinking about this, as I mentioned, was this other project with another PhD student. So he now has a teaching postdoc at NYU. And so in this project, we were looking at the number of unique topologies. So this was data sets of 500 gene trees and either 5, 10, 15, or 20 taxa. Lambda here was the speciation parameter for a Yule model. And basically, the larger lambda is, the more rapid speciation is. So that means the shorter the branch lengths are, and that means in the coalescent sense, the more incomplete lineage sorting there is. So the more variability there is in the gene trees, and so you expect a higher number of unique gene trees. Okay, so with a set of 500 gene trees, at least by the time you get to 15 taxa, there's a reasonable chance at higher levels of incomplete linear sorting that all gene trees would have a unique topology. And for 20 taxa, it's most often the case, at least for higher levels of incomplete linear sorting, that all gene trees would be unique with 500. And so this is kind of the motivation for thinking that comparing the number of observed topologies to the number expected might not be very informative, because those numbers are, the topology counts are just going to be zeros and ones most of the time. Okay, and then there's other things to do here too. This plot is just based on true gene trees that are just generated under the multi-species coalescent. So if you have gene trees that are estimated from DNA sequences, then that adds noise. So the proportion that are going to be unique is going to be higher than what's observed here. And we did some of that. And for the cross-validation study, we're in the process now of doing this with estimated sequences. And so with estimated gene trees. So again, with estimated gene trees, it's going to increase the number of unique topologies. And I think that's going to give more motivation to using a triplet-based summary or some sort of summary of the gene trees, instead of using gene trees that are that are likely to occur uniquely. Okay. Yeah, so in conclusion, the tripletized cross-validation had similar performance to using full gene trees, at least a smaller number of leaves, slightly better performance with larger numbers of leaves, and it also runs faster, just computationally. But my main motivation for thinking about it is this problem that you can easily have data sets where all the gene trees are unique. Okay, so unfortunately, trying to run PhyloNet with more than 9 leaves, it often will crash or have memory problems getting the full likelihood. So if I wanted to try this for larger networks, I'd I'd probably need to use branch lengths in the gene trees, which it's much easier to calculate the likelihoods in that case. Yeah, and then I'd have to worry more about sensitivity to errors in the branch lengths. So anyway, so that'll be ongoing work. All right, are there any questions?